These are the lecture notes for Chapter 28 on invertebrates, which are animals without a backbone. In Section 1, there's a little bit of information on the evolution of animals and characteristics that all animals have in common. And then Sections 2, 3, 4, and 5 go through the different types of invertebrates that exist. So we'll divide those up into the different phyla. So first of all, make sure that you know the characteristics that all animals share. If an organism is a member of the animal kingdom, then it is multicellular and heterotrophic, which means it must consume its food, and animals ingest their food, whereas fungi, as you know, absorb it. So animals ingest their food and digest their food internally. Common features of animals, these slides just show up here in picture A, these are neurons, nerve cells, in other words, and picture B, of course, we're looking at someone having a meal, and then we've got the cheetah, fastest animal on earth, and then what you see in picture D is a ball of cells that formed as the zygote, the original cell, the fertilized egg, began dividing. So this is very, very, very early embryonic development. Now, on this page, I just want to remind you that it is thought that the ancestor of animals is a single-celled protist, similar to a choanoflagellate. And a choanoflagellate is a protozoan. It is single-celled or unicellular, and it has a flagellum. And it looks kind of like this. Okay. Now, when we talk about the animals, we need to talk about the different types of body symmetry, because symmetry is used as one of the characteristics to kind of group animals in different different phyla. So symmetry is a pattern of similarity that is observed in objects. There are three types of symmetry. There's, first of all, asymmetry, which is no symmetry. The sponge is the only animal that is asymmetrical. So that's no symmetry at all. Then there is radial symmetry. That means that you could take the animal and cut it into two identical halves in more than one way. So think about a jellyfish. I could make a vertical cut or a horizontal cut or a diagonal cut and get two identical halves. But I could not do that, for example, with a human or with an insect. Think about a grasshopper, which has bilateral symmetry. That means there are definite right and left halves. You can cut the animal in one direction and get symmetrical right and left halves, but not like you can with jellyfish, where you can make different angles of cuts and get symmetrical halves. So in bilateral, there's definite right and left halves. The animals tend to be active. They tend to move forward at the anterior or head end, and they do tend to have cephalization, which is the development of a head region with possibly a brain. Now, with embryonic development, the first tissue layers are called germ layers. And the germ layers give rise to the organs and the organ systems. If an animal has no germ layers, they have no tissues, like a sponge, for example, the sponge literally has no tissues. It's got different cell types, but it doesn't have any, technically any tissues. But then you can have animals that have only two tissue layers. That would be an endoderm and an ectoderm. These animals are going to be our jellyfish and our comb jellies. They'll be the second and third animals that we learn about after the sponge. And then the rest of the animals in the animal kingdom are triploblastic. That means they have three tissue layers. They have an endoderm, which is on the very inside, and then they have an ectoderm on the outside, and then they have a mesoderm in the middle. So with the triploblastic animals, they can be divided into protostomes and deuterostomes. The only deuterostomes that are in this chapter, the invertebrates, are the echinoderms, which include the sea stars and sand dollars. So they're the echinoderms, like sea stars, are the only deuterostomes that are invertebrates, and the rest are going to be in the vertebrates, which is our next chapter. Now, the main difference is that these triploblastic animals, as they develop two openings. And the openings are, for the digestive tract, for the GI tract. So there's a mouth and an anus. Now, if that first opening becomes the mouth, then it is a protostome. But if the first opening becomes the anus and the second opening becomes the mouth, then it's a deuterostome. Protostome means first opening, and deuterostome means second opening. I think protostome means first mouth, and deuterostome means second mouth. Anyway, the protostome, the first opening is the mouth, and the deuterostome, the second opening is the mouth. Okay, so radial symmetry, what you're looking at here is a body form that's very similar to a sea anemone, which is in the same phylum with the jellyfish. And you can see the glass is making different cuts in different directions. We have two diagonal cuts and then a horizontal cut. And any way you divide this organism up that way, you're going to get equal halves. But with the turtle, the sea turtle here, or tortoise, I'm not sure which one, you can only make a cut in one direction to give you two symmetrical halves. This animal has obvious right and left halves. That's bilateral symmetry. Humans are the same way. And also, animals with bilateral symmetry tend to have a head region, and we call that cephalization. As far as directional terms, the stomach region is ventral, and the front region is anterior toward the head. The back region is dorsal, and then the tail end or lower end of the animal is posterior. Head end is anterior, and tail end is posterior. Back is dorsal, stomach is ventral. All right. When the original zygote, the zygote is the fertilized egg that has been fertilized by a sperm. It is the first cell that develops into the adult animal. So it's the very first cell. It's called a zygote. When that zygote begins to divide, the overall size of the zygote, as it divides, it doesn't get bigger. In other words, when it divides into two cells, each of those two cells are half the size of the zygote. So we call this cleavage, cell division without growth. And what you were looking at back in this picture here, this represents cleavage. You see all the individual cells, they'll divide more, and as they divide, the new cells are smaller. They're half the size of the parent. So this entire ball of cells is the same size as the original zygote. And that is cleavage when that happens. That happens early in development. Protostomes undergo spiral cleavage. Deuterostomes undergo radial cleavage. I honestly, myself, don't 100% understand that. I think you would have to watch it, maybe with a video, like a time-lapse video. But you do need to know it, that protostomes undergo spiral cleavage, and deuterostomes, the cleavage is what we call radial. As the zygote undergoes cleavage, it first forms a ball of cells called a blastula. The protostomes, the first opening of the blastula becomes the mouth, and deuterostomes, first opening is anus, second opening is the mouth. Now, some protostomes have a body cavity called a coelom, but all deuterostomes have a true coelom. And the coelom forms in two different ways. If the protostomes have a coelom, it forms by a splitting of the mesoderm tissue, that tissue that forms kind of in the middle between the endoderm and the ectoderm. And deuterostomes, their coelom forms by an outpocketing of the primitive gut or digestive tract. And here you can see a comparison of protostomes and deuterostomes. So let me blow this up. In protostomes, you can kind of see what is meant a little bit in the picture by spiral and determinate cleavage. That's what protostomes have, is spiral cleavage, and it is determinate. And then deuterostomes have radial and indeterminate cleavage. So the best thing here, honestly, is just to memorize that information. I can explain the rest, but to me, it's just kind of difficult to understand, and you really just need to see it in like a video more than just a picture doesn't really explain it that well. Protostomes. Okay, you can see the three tissues here. You can see the outside ectoderm is colored blue. The endoderm, that's going to become the lining of the digestive tract, is colored kind of a beige orange color. And then the mesoderm is pink. And you can see the first opening, called the blastopore, becomes the mouth. In deuterostomes, that first opening becomes the anus, and the second opening becomes the mouth. They look very similar in the end as far as at this stage, but notice that the mesoderm in the deuterostomes formed as an outpocketing of this endoderm tissue, the primitive gut. All right, and then we have the formation of the coelom. So the coelom is the body cavity that holds the organs. So in the protostome, you have the gut, which is lined by endoderm tissue. And then you have the mesoderm here and here. And the coelom actually forms by a splitting of the mesoderm. The coelom is not labeled, but this is the coelom. Just this space. The space between the ectoderm tissue and the endoderm tissue. All of that space in here is the coelom. And then the coelom, I mean, they end up looking the same, but the coelom in the deuterostome forms a different way because the mesoderm forms a different way. So mainly, just to make sure that you know, the primary differences are more than likely what the only thing you're going to be asked is going to be that protostomes, the mouth forms first, and deuterostomes, the mouth forms second, the anus forms first. Those are most often the questions that are asked. Now let's move on to the animal phyla. The very first animal phylum, which is the simplest, is called the Porifera, because the sponges, which are the only animals in phylum Porifera, do have pores. So sponges are actually animals. They lack true tissues and have only the cellular level of organization. They only have a few cell types. One of their cells is actually similar to the choanoflagellate protozoan that we looked at earlier. They have sac-like bodies that contain many pores. They have flagella in those cells called choanocytes. I think that's what they're called, choanocytes. Do you see that anywhere on here? Yes, called collar cells or choanocytes. And they have, basically they are filter feeders. They are sessile as adults. All animals have movement at some stage in their life. So the sponges only can move when they are larvae. But as adults, they are sessile. They're attached to something and they just remain in place. They filter feed water that flows through their pores into a central cavity and out their osculum, which I will show you. Water flows in through the pores and then up and out the osculum. For their reproduction, they reproduce sexually and asexually. Asexual is by fragmentation or budding. Sexual reproduction produces a flagellated larva that can swim to a new location, attach, and grow into an adult that is sessile. Sponges are capable of regeneration. As far as the cells that form a sponge, you have these collar cells that filter food into the sponge. You have these amoebocytes, which is where digestion occurs. And this is a yellow tube sponge, the one in the picture, in picture A, called a Plycina fistularis, but it's just a picture of a sponge so you can see it. You can make sponges, commercial sponges, out of the animal, the dead animal. The dried body of the dead animal can be made into a commercial sponge, but they actually make synthetic sponges mostly. They don't normally make them out of the actual animal. So next recording, we will start with phylum Ctenophora. The C is silent.